So generally, we can classify the protein into these two main classes, okay, fibrous protein and globular proteins. But please do bear in mind that we can classify protein based on their role. For example, if form the receptor, we call them receptor protein. If they form hormones, we call them hormonal proteins. Okay, if let's say they form we call this involved in the biochemical reactions, the enzymes, so we call them as enzyme. Okay, so if they form these structural roles, for example, they maintain the shape of our face, for example, firm our skin, right? Collagen particularly, then we call them as the structural proteins. Okay, so we have many groups. For example, immunoglobulins, our antibody, we call them as a defense protein. So it depends on whether you want to classify them based on the shape or based on the role. So for today, because we don't go into the detail about each kind of proteins, we generally divide them into two main classes based on their shape. So one is a fibrous, means fiber, they form fiber, long chain. Okay? Another one is called globular. Globular means spherical, round shape. Okay, they form sphere. Okay, so you can see that A, this case, is the fibrous protein where we refer to the hair keratin. Okay, so what is the property of the fibrous protein? We're going to look at it later. And globular protein, basically, they form a sphere, spherical shape. So how they can form this spherical shape? We're going to learn them again. Okay, so example of the fibrous protein, okay, keratin, collagen, they are fibrous protein. Globular protein, for example, enzyme, hormones, hemoglobins, those are globular proteins, okay, because they were curled into or form this what we call the spherical shapes. Okay, so let us compare between these two, okay, to find out the differences between the fibrous protein and globular protein. Example of the fibrous protein, keratin and collagens, they are well-known fibrous protein. Okay, and how about the globular protein? Globular proteins actually enzyme, some water-soluble hormones, hemoglobins, they are globular proteins. Okay, now how about in terms of structure? Fibrous protein, in terms of structure, they are long polypeptide molecule, lie side by side. Then very important, they tend to form what we call the fibers. Okay, they form fibers, so they are long chains. Okay. But how about globular protein? Globular proteins, they tend to curl up into the ball shape. This is very important. When you curl up into the ball shape, because they try to protect this non-polar hydrophobic R group. So those they don't want to interact with water, what they do? They hide themselves at the center core of the proteins. For example, you curl into this already. So at the center core here, we do have those R group that don't want to interact with water, so they're away from the water surrounding aqueous. Okay, and how about polar? Polar hydrophilic R group will stay outside. So those are hydrophilics. So it means that they can interact with water molecule. So when they interact with water molecule, we say that they maintain the solubility. Okay, they maintain the solubility of the protein. So it means that this protein won't form aggregates. They won't stack together, they don't join together, but this protein was exist as the single molecular form. So we say that they dissolve. Are you clear? They dissolve in water. So protein, we have the water-soluble form, globular protein. Why? Why they can solubilize in water? Okay, basically they have this hydrophilic R group forming hydrogen bond with surrounding water molecule, therefore they dissolve. Are you clear? But fibrous protein, they don't dissolve, okay, in the water. So that's why you say insoluble in water. If they're soluble in water, then we're going to cry off, as well will be no hair. You try to imagine your hair, the keratin can dissolve. You don't have fingernail, you don't have the hair anymore. You try to imagine every time you wash your hair, oh, your hair soluble in water, gone, no more. Okay, hair you have, or we have. Okay, so that's why fibrous protein, they are insoluble in water because they hardly form hydrogen bonds with water molecule. But globular protein, yes, they're soluble in the water. So in terms of function, fibrous protein normally give us a structural role. So what means structural role? maintain the structure of our, okay, for example, to keratin, make your hair, your structure of the nail, okay. For example, we have collagens, we take collagens, right? So collagen actually help us to firm our face, firm our skin, right? So structural role, give us the structural, okay. But globular protein mainly involved in the metabolism. For example, the enzyme, our hormones, okay, involved in the metabolism. Our hemoglobin transport oxygen, right? So involved in the transport. Are you clear? Transportation role here. So let us continue with the collagens, okay? So collagens, they are fibrous protein, okay, found in the skin and the wall of the blood vessel. So now I need to introduce you guys with three terms, okay? Be careful. We have collagens polypeptide, or sometimes we call it a collagen chain, okay? Then we have the collagen molecule, and as well as collagen fiber. So you need to understand these three, okay? So collagens, when you say use the word collagen, actually we refer to collagen fiber, okay? So they are fibrous proteins found in the skin and the wall of the blood vessel. So skin, to make sure that your skin is firm, it won't saggy, I mean your skin, okay? And the wall of the blood vessel, because we don't want our blood vessel to burst when it's under pressure, okay, or overly stretched. So one collagen molecule will have three polypeptide chains. So how it look like? So first of all, we have one collagen chain of polypeptides. Can you see that? So three of them, their helical structure, three of them will link together to form one collagen molecule. Okay, so this is collagen polypeptide. Okay, three of them join together, then form one collagen molecule. Okay, and collagen molecule join together to form collagen fiber. Are you clear? So can you differentiate collagen polypeptides, collagen molecules, and collagen fiber? So if you understand these three, then we can continue now. Okay, so first we have three of them, okay? So these three helical chains will wind around to form a three-stranded rope-like structure. And how we link three of them? By hydrogen bond. So means that collagen, the collagen molecule here is termed as a quaternary protein. Why we call that as quaternary protein? Because they consist of more than one. They consist of three polypeptide chain joined together. So how we join them together? By the R group interaction forming this hydrogen bond. And something special about this collagen polypeptides, every third amino acid is glycine. And we know that glycine is the smallest and simplest amino acid. So when they are small, so they will be able to make these three, okay, small molecular mass of glycine allow the three strands to lie close to each other. Okay, you try to imagine that if you have something small, every three of them, they are small. So means that at this small region part there, they will be able to wind together, okay, and then allow the hydrogen bonds to be formed between two polar R group, not between two glycines. Yeah, glycine won't form hydrogen bond. Okay, glycine are non-polar. Okay, so between polar R group, the hydrogen bonds can be formed. Okay, so try to imagine that if I use all big one, okay, without small one, so it's very difficult for you to wind. You try to imagine that you want to intertwine those big chain, difficult. Right? They will, I mean, the interaction won't be strong. But if you have something small in between, then you can put another chain here, a small part. Then you can do wind at the small parts. The what we call this the small regions there. You will be able to wind them nicely. So that's why collagen molecule can be formed by 3 intertwined, okay, collagen polypeptide, okay. And we do know that collagen fiber, they have very high tensile strength. So tensile strength because they coiled, okay, and these 3 collagen molecule will interact with other collagen molecule. All right, these 3-stranded collagen molecule interact with other collagen molecule running parallel to it by forming covalent linkage. Can you see that? So can you see that? 1, 2, 3 of them, they link. But now they link with the covalent bonds now. Okay, so between this, within one collagen molecule, they link by hydrogen bond. But between some collagen molecule, they form covalent bonds. So hydrogen bond, covalent bonds. Are you clear? Again, hydrogen bond, covalent bond. And they stagger like this. So making them have high tensile strength. Are you clear? Okay, so they interlock something like this. They stagger like this. Can you see that? Rather than they make it this way. They make it this way, they don't have tensile strength because when I exert the pressure, they were slippery. Okay, but they arrange this way, 3 of them. And then another, okay, join, and then like interlock. Can you see that? They interlock each other together. So therefore, they have very high tensile strength. So how we join them together? This one's by covalent bonds, okay, for collagen molecule. Okay, so this one is called collagen fiber. This one called collagen molecule. Okay, and this one is the collagen polypeptide. So 3 collagen polypeptides join together or link together by the hydrogen bond, and then polypeptide molecule join with another polypeptide molecules to form fiber by using the covalent bond. Okay, so the next proteins that we are going to look at it is hemoglobins. Okay, hemoglobins. So hemoglobins, we do know that we learned it. Okay, they are the globular proteins, means that they're soluble in water. They are oxygen carrying pigments. So if we're zooming into hemoglobins, they consist of 4 polypeptide chains. 4 polypeptide chains. So we have 2 alpha and 2 beta. Now be careful. 2 alpha, 2 beta doesn't mean that they have alpha helix or beta pleated sheet. It's just that because 4 of them, 1, 2, 3, 4 of them join together, and then we give them a name. And so happen that 2 of them, they're identical. So that's why we name them 2 alpha, 2 beta. So nothing to do with alpha helix or beta pleated sheet. Are you clear? Okay, so it's just a name. For example, okay, we do have the person named as Jamie Foxx. So the fox doesn't mean that it's the animal, right? So it's just a name. So alpha helix and beta pleated sheet here doesn't mean that they must have alpha helix or beta pleated sheet. It's nothing to do with alpha helix and beta pleated sheet. It's just the name we give to the polypeptide chains, okay? So how we can pack them together? So these 4 polypeptide chains are packed closely together so that the hydrophobic R group always point toward the center, okay? Because why? Hydrophobic don't like water, right? So we make them at the center. So when they are center, this hydrophobic R group help us to maintain the precise 3-dimensional shapes of the proteins, okay? So hydrophobic R groups at the center help us to maintain the 3-dimensional shape of the protein. Correct or not? You try to imagine that you fold, okay, something. If you didn't use the glue to glue them, they will unfold. Correct or not? So this hydrophobic R group help us to maintain the shape. How about hydrophilic R group? Hydrophilic R group usually point outwards so that it can interact with water molecule to maintain the protein solubility. So if the protein solubility decrease, what will happen? It tends to precipitate, so become functionless. You try to imagine that, okay, every time we add sugar into a drink, until a moment when you add the sugar, it no longer dissolve, it precipitate. So when precipitate out, then it won't have the functions anymore, okay? And each polypeptide chain contain the heme groups at the center there, heme heme groups, heme groups, and heme groups. So this heme group actually is called as a prostatic group. So what does mean prostatic group? Prostatic group is a permanent part of protein, but they are not made of amino acid. So they are proteins, they are non-protein structure that attach to the protein so that they enhance the functions of the protein. So prostatic, okay, think about this prostatic leg. Prostatic leg, okay, is a part of your body, but they are non-cellular structure. Same idea here. They are non-protein structure, but they are part of the protein. Okay, so this heme group contains the iron ions, Fe2+. So these iron ions actually can help us to transport oxygen. And how we transport oxygen? So we do know that one hemoglobin's molecule consists of 4 heme groups. Each heme group can take 1 oxygen molecule. So therefore, one hemoglobin can take 4 oxygen molecule to form oxyhemoglobin, HbO8. So total 8 atoms of oxygen can be carried by 1 hemoglobin. Okay, so 1 hemoglobin can carry 8 atoms of 4 oxygen molecules. Okay, be careful when you write. Okay, so this is the structures of a hemoglobin. So you can see that we have beta 1, beta 2, but both of them, they are identical. And then you have alpha 1, alpha 2, they also identical. So each of them, they have this heme group. So heme group at the center of the heme group, we have the ion. So this ion can be converted, can we say that? Interconverts, okay, interconversion between the Fe2+ and Fe3+. Okay, so your blood, when it receives oxygen, so why our blood is red color? Because of Fe3+. Okay, but when deoxygenated, it's a bit greenish in color because of Fe2+. Deoxygenated, right? In the blood vessel, deoxygenated, they look like a bit greenish in color, right? Okay, because of the Fe2+. Okay,